they also give you more insights into key biological questions. So this is a nice example from work from the Allen Institute in Seattle, where they did profile cells from different species. So they looked at mouse and human, for instance, and then they compared cells from the cortex. And if you know a bit about literature about cell types in the brain, you know, for instance, that cells in the cortex, the outer part of our brain, are organized into layers, and cells are layer specific, right? And this is the picture you see at the bottom here. What we didn't know, and that we know now, is that if you look in the human brain, the picture is quite different, where you see that cells of the same cell type, they actually span multiple different layers, right? So this data allows you to see something that we actually didn't know before. More importantly, and also more related to translational applications, is differences between gene expression. So here, this is an example that they showed in the paper of serotonin receptors, and these are very well-known targets of antidepressant drugs, for instance. And what you see here on the different columns are the different cell types. And just overall looking at this picture, you see that the same receptor is actually expressed in different cell types between mouse and human. So if you develop a drug and test it in mouse, you should be aware that the effect in humans will be quite different because you're targeting a different cell type. So what I want to focus on today is some of the computational challenges that we get from this data. So I tried to summarize this a little bit into challenges that come from the fact that we have now the ability to measure from different individuals. So we're building atlases, for instance, or we try to look at EQTLs at the single cell level, or we just simply want to compare groups. And the challenges there come from the fact that, well, the amount of data is continuously growing. So how do we deal with that? People annotate cells differently. And the second point would be about using multiple modalities. So we can now measure mRNA. That has been a lot of push in that direction, but we measure also accessibility or spatial data. And most of the time, these technologies are destructive. So even if you can measure two of these properties or features in one assay, usually you cannot do more. So if you do more, then you would need a way to merge or integrate your data. These features are not matching. So with accessibility, you can indeed reduce it to the level of the gene, but you do measure accessibility across the whole genome. So how do you merge this with mRNA, for instance? And the last point I will not have time to talk about today, but like the example I showed you, there is a lot of interest in studying, for instance, evolution of cell types or comparing cell types across species. How do we actually do that, right? From the beginning, the sets of genes that we have in these species are quite different. So how do we get to a point where we can match cell types and say this is exactly the same cell type that exists in two species? So, okay, starting with the first one. Over the years now, people have been collecting these data sets, and eventually we would like to end up with some sort of an atlas that combines all these data sets for a specific tissue. And to get there, there are a lot of integration methods, right, that try to get rid of these batch differences and so on. But assuming that we have this atlas, it will be very nice to use it to train some sort of a model, a classifier that can learn the separation between the different cell types. So if we collect a new data set that's not annotated, we can automatically get the labels of this data, right? This would save us time. It will make things a lot more standardized across different analyses, and will make it much easier to compare different data sets. But there are some problems actually, and from what we have done before, the problem is not really in the classifier or in the model. This we can tune this quite effectively. The problem is that this atlas does not exist yet, even if there are a lot of data sets available. There is no complete atlas, and there are many nice efforts ongoing on constructing a human cell atlas and so on, but we're not there yet. So we're not there yet, and we might actually never be there, right? Because to build an atlas for a specific tissue or an organ, you do need to cover a very wide range of variations, right? So we might not have cells collected from every single disease that's out there. Or think about complex tissues like the brain, for instance. There are multiple brain regions. Cells differ across the development stages. Again, across species, there are marked differences. So this atlas might be a bit difficult to get to a fixed point. There's a problem that's very practical: inconsistent labeling. So we don't know how to call cells. This is an example dendrogram of cell types from two big studies that came out in the same year. And what you see here is that one group really focused on labeling these excitatory neurons, for instance, based on which layer they are in, while here they label them differently based on which peptides, for instance, are expressed and so on. So how do we match these labels across different studies is a big question. Different groups are interested in different cells. So you can take this very, very simple example. If you're interested in immune cells, one group might be sufficiently satisfied with labels annotated as T cells, while others would like to go a little bit deeper into separating CD4 and CD8 T cells. So how do we kind of get automatically that these cells actually belong to the same group? And the last point is that a bit related to the first one is that we would like to always be able to flag or label cells that are unknown, that we have not seen in the atlas before, because these would be the interesting cells, for instance, that are related to a new disease that we're studying or something like that. So our solution to this is to develop scHPL, which is single cell hierarchical progressive learning. And the main aim here is to take annotated data that exists already publicly. We can build a cell hierarchy out of this existing data that we can then later use to label new data sets that become available. So I'll try to walk you through how it works. So let's take this example where we have two data sets. The first one has 3 populations, this has 4, and just based on this schematic, we know that A and B should be subpopulations of 1. So in a way, this is kind of the tree that we want to construct at the end. The way we do it is that we train a classifier on dataset 1, and we call this flat classifier because there is no hierarchy taken into account here. And we do the same for dataset 2. And then we use these trained classifiers to cross-label cells from the other dataset. And in this way, cells that belong to population 1 over here will be labeled as A and B. So half of it will be A, half of it will be B. And A and B over here will be labeled as 1, right? Because this classifier that was trained on dataset 1 only knows label 1, doesn't know A and B. And using these classifications, we have a scheme to match the labels. I will not go into the details of this, but we can basically end up reconstructing this tree quite easily. If we now add a third data set that has a separate label, so for instance, we figure out that actually C and D come from a larger population over here, then we can add this population to the tree, and then C and D or 2 and 3 become subpopulations of this super class. Yeah. And eventually, at some point, we have added all the data sets that we would like to add. So then we have this final tree. We train the classifier on every node of that tree. So we perform hierarchical classification. So if we get a new data set, then we can automatically label these cells with the option to flag new populations that we have not seen before. Yeah. Okay, so does this work? I have here one example, again, on brain datasets. So what you see on the left is a UMAP of integrated data from 4 different sources. I'm showing you 3 of them over here that we will use to build a tree. And then the 4th one, the Rosenberg data, we will use as a test data in this case. So what you see here is that these 3 atlases, the 3 datasets, they basically have a varying number of cells. Also, the annotations are quite different. So I zoomed in here on one example. So these are labeled as vasculature cells in the Zeisel data. Here, some of them are brain pericytes, some of them are endothelial cells, and here they are called endothelial stalk, endothelial tip, and mural, and so on. So in the first iteration, we add the Saunders and the Zeisel data sets, and then this is the tree that we get from scHPL. And what you see here is that these 3 populations from Saunders are added as a subpopulation of the vasculature population. Yeah, so this seems quite right. And then if we add the tabula moris data, you see here that the endothelial stalk, endothelial are matched, and the same for mural and brain pericytes. And they are all still subpopulations of this vasculature population from Zeisel. So this is kind of the relationships that we want to capture, basically. You see it here in the UMAP. We basically want to capture this automatically, and it seems scHPL is doing this quite okay. Now we train a classifier on this resulting hierarchy, and we test the labeling of the Rosenberg data. So these are the original labels from the Rosenberg data. What you see is that they have a very detailed level of annotation over here. These are the labels that we have, so that's what we can predict. So for instance, these are all neuronal populations. You see that the original data annotated at a much deeper level. They are all now subpopulations of neurons in our case. And in particular, you can look at neurogenesis, for instance. We only have a general neurogenesis label, but that's really capturing all these precursor or progenitor states that were annotated in the original data. So we think this is working quite nicely. There is a catch, though, as in I told you that we would like to use this to be able to handle growing data sets, right? But the first step we did is that we integrated all the data. That means that if we get a new data set now, we need to repeat everything that we have done because we need to reintegrate the whole data set. And that seems a bit of a problem, and indeed it is. So last year, we saw this nice work that came from the lab of Fabian Theis from Munich, in which they developed this scHPL. So they use surgery to basically, they start with a set of data sets, they train a model. In this case, they use a deep learning model like SCVI, for instance. So you can learn a latent representation of the cells. And here you have a label for the batch from which the cells come from. What's nice about this model is that if a new study comes along, you can basically add this the new data, and you don't need to retrain the whole network. You don't need to relearn the latent representation. All you need to do is to update these bold weights over here. Yeah, and that sounded like a very nice solution actually to the problem, where we can have this kind of integrated data. We can use scHPL on that. On Z, on this latent representation. And if a new data set comes along, we can just add it and then get an updated Z that we can use later in scHPL. So we don't have to throw away everything that we have done before that. So yeah, we're working together with them at the moment to have a new method that combines both. So I hope we can have that ready early next year. So just to summarize this first part, scHPL basically can take 2 or more annotated data sets, and then it will basically give you a cellular hierarchy, which can be interesting on its own. So you can use it to learn actually what's the full picture of cell hierarchy in that tissue. And then we give you also a classifier that can be used to annotate new unannotated cells using this hierarchy that we have learned. At the moment, I did not discuss this, but the classifier is really, you're really free to use whatever. In our implementation, we have different versions of support vector machines that are better or worse at handling rejection, so the annotation of unknown cells. But yeah, the model itself can also be changed in that sense. So you can use any classifier if you think it's better. We can update the hierarchy, and the good thing is that we can always preserve the older annotations, right? We don't throw them away. So if you have done any inference on these older annotations, like looking at single cell QTLs or something, we can always go back to the original annotations. We don't throw this away. And as I mentioned, the major limitation is that we require the data sets to be pre-aligned before we start. And we hope this integration with scHPL would overcome this limitation. Okay, moving on to the second part on the multiple modalities. As I said, it's very interesting to look at different aspects of the cell molecular state, but a lot of the technologies are very destructive. And the one example I want to focus on here is when we measure single cell dissociated measurements and also do spatial measurements. So in this case, it's about oncology. So you have a tumor, you can dissociate the cells, separate them, and then you can do either mass cytometry or single cell RNA sequencing. The nice thing about this is that you measure, let's say, if you go to sequencing, you measure the whole transcriptome, right? The bad thing is that you lose completely any info about the spatial location of the cells. And for tumors, for instance, the microenvironment is very important. So that might be something that you want to look at. From the other side, there are methods now to do spatial transcriptomics. So you can measure transcriptomics or gene expression on the tissue itself. So of course, then you retain the spatial information, but there are some limitations. So some of the protocols are limited in terms of the number of genes that you can measure, or they are not at the single cell resolution, like Visium, for instance, from 10x that gives you these spots, and these spots cover multiple cells. And some of them are limited by their availability or cost, for instance. That makes it really difficult to apply these spatial technologies. So in many cases, it's important or it's beneficial to integrate spatial data with single cell data. And two of the major applications there are, the first one is about predicting locations. So if we have some prior data, prior spatial data for our tissue of interest, and now we collected a new set of single cell data that's dissociated, can we predict the location of these cells in the tissue itself so that we know which cells would have been at a specific location? And there are multiple nice models out there. So NovoSpark, cell to location. We're also working on a new model to do this more efficiently. But the other thing you might want to do is that you get a spatial data that doesn't measure all the genes, only a subset of the genes. And you have a single cell data that comes from the same tissue as well. And by integrating the data, you can predict the expression of genes that you have not measured spatially. So you've only measured them in the single cell data, but not in the spatial data. And for this, again, there are multiple models. We developed SpaGE, so that's what I'm going to explain today. Okay, so just to reiterate, we have single cell data in which we have cells, they are dissociated, no spatial info, but we have measured all the genes, right? So the whole transcriptome. And on the other hand, we have a spatial data where we have measured some genes. So let's say between 10 and 1,000 genes, depending on the protocol. It comes from the same tissue, right? And at the end, we want to integrate them so we can get the spatial profile of all the genes, right? Measured ones plus the unmeasured ones that we can predict. An easy way to do this is to take any spatial cell, so let's say the blue one, and then you look for similar cells in the single cell data, the red ones, and you do this based on the shared genes. So assume that you have measured 1,000 in the spatial data, you use these 1,000 genes to get distances and look for neighbors. And once you have found the neighbors, you can just take the median or the average of their expression of the other genes, right? Because for the red cells, you have the measurements of the whole transcriptome, including genes that you have not measured for in space. There is one problem with this approach, and that is these data sets, they don't live in the same space, right? So we do need some sort of alignment or integration in between them beforehand. For this, we rely on a method called Precise that performs domain adaptation, and this was developed by Sofian Moragi. So he's a PhD student in Delft and in the NKI with the Lodewijk vessels. And the method is relatively straightforward. So you perform principal component analysis on, here we call it the reference data, that's the single cell data. And we perform principal component analysis on the query data. So in this case, it's our spatial data. And then what we do is that you basically perform singular value decomposition on the cosine similarity matrix of these two dimensionality reductions. And the aim of this is that you actually align these principal components together. So trying to put this visually, assume that these are kind of the two spaces that we're interested in. So this is principal component 1 and 2 of the single cell data, and this is for the same for the spatial data. By doing this, you applying Precise, you actually define these principal vectors that are kind of aligned. So if you look at the cosine similarity between the principal components of the single cell data and the spatial data before alignment, there is actually not so much similarity in between them. After alignment, you basically reorder, you rotate them in such a way that the first principal vector of the single cell data aligns quite well with the first principal vector of the spatial data. Yeah. And it's in this space that we are going to do this KNN imputation and look for neighbors to do the prediction. Okay, so let's see if this actually works. So this is an example in which we are looking at the primary visual cortex of the mouse. These are two data sets that profile this region from two different studies. So this one is spatial data measured using the STAR-MAP protocol. There are 1,500 cells in this data. They have measured around 1,000 genes. And the single cell data is from 14,000 cells, and you get measurements from all transcripts. So what we're going to do is that we're going to take genes that are measured in the star map data. We mask them as if we don't know their expression pattern. So here, for instance, this is the measured pattern of COX-2. And then we use the remaining genes to align the data and do the prediction. And then we look at what is the predicted pattern of COX-2. And in this case, we get something that looks very, very similar to the measured data. Yeah? So it's sort of a leave one gene out validation. Of course, we can do this for every gene. So here I have 5 carefully picked examples where we show you that the measured expression and what we predict actually matches quite well. Even for genes like BSG that have a very sparse expression, for instance, over here, you see the pattern from the predicted from SpaGE is quite similar. Yeah. Okay. We can quantify this, and we relied here on the Spearman correlation of the predicted and the measured values. And you see that overall these correlations are super low. We can discuss this later. In short, we don't think correlation is the perfect measure, but it is a measure of how good this prediction is. And if we compare what SpaGE is doing to other methods here at Leiden and GimVI, we see that we are slightly but significantly doing better overall. One thing that we were interested in is that there were some genes that we knew, for instance, they had an interesting layer pattern across the mouse cortex, and that you see here in the prediction. But when you look back at the measured data, it's actually not very clear, right? So this is, for instance, SIRV. We know very well that this should be expressed in this layer. There is a bit of signal here, but it's not very convincing. So in that case, it seemed that the predictions are actually better than what we get from the measurements. And if we go to another data set, another spatial data, OSM fish, so this is cyclic fish measurements, you see that indeed what we predicted here aligns with what we see in the OSM fish data. I have to say this is a different region of the cortex, but still the layering structure does not differ too much in between these regions. Okay, and now we were convinced that, okay, we're doing good measurements. So can we actually predict genes that we haven't measured at all? So these are 2 examples. So these are genes that do not exist in the star map data. So we don't have a good way to validate them, but to us, they looked quite nice, right? They are expressed in specific layers of the cortex. So if we go back to the Allen Brain Atlas, which has measured the expression of every single gene in the mouse brain using in situ hybridization, so it's not really single cell level. But again, if we zoom in on the same region, so this is the primary visual cortex, you see that the expression of the genes that we have predicted aligns very well with what we see in the in situ data. Yeah, so we're quite confident that you can actually make predictions on the spatial expression profile of genes that you have not seen, you have not actually measured. I'll show you one more example in this case. So this is MERFISH data. In this case, we have way more cells. So this is around 64,000 cells. We have measured less genes, 155. This comes from this study. And actually, there is a matching single cell data from the exact same paper. And again, a bit of the same story. If we make these predictions, we're slightly better than other methods, maybe even the same here. So really, really small differences. But where the differences comes in is in the simplicity of the model, right? So CERAT relies on canonical correlation analysis, for instance. Liger, NMF, non-negative matrix factorizations. GimVI is a deep generative model. And in this case, SpaGE is very simple, right? You do PCA, and SVD, so it's a very linear method, can be pretty quick. So in terms of memory on the y-axis here and computation time on the different data sets, so in the black colors, you see you see SpaGE. It's way more efficient than all the other methods that we compare to. Okay, in the last bit, I want to show you what we are doing at the moment. So what we thought of is that with SpaGE, we predicted these expression patterns that we have not measured, right? But there are a lot more features that you can get from the single cell data that are not available in the spatial data. And one of those things are these RNA velocities. So if you're not aware of this, It's a paper that came from Sten Linnarsson's lab a couple of years ago, where they made the observation that if you look actually at single cell data, you do expect that all the reads that we detect are coming from mature RNAs, right? We're profiling mRNA from the cytoplasm. But what they saw is that there is a varying percentage in red over here, so around a quarter in the 10x data, that comes from unspliced reads, right? And they thought this was very interesting. And the way they made use of these reads is that they linked them to RNA metabolism. So if you look at this kind of simplistic model, you have transcription, you make unspliced mRNAs, then splicing happens, and then you end up with a spliced mRNA, and then degradation happens, right? So if you look at it over time over here, the rate at which you generate these unspliced mRNAs and then degrade them is kind of a little bit ahead of the splicing, right? You see this curve and this curve are a little bit delayed than the blue one, right? So if you look at it in this space, so if you look at unspliced versus spliced, you kind of go around this cycle where and at the moment where the cell is transcribing the gene, you actually make more unspliced reads than spliced reads. And then at some point when you're degrading, there is more spliced than unspliced. Yeah? And you can do this for every gene, basically. And if you average across them, then you can end up with these small arrows that tells you, okay, it seems that this cell is going in that direction, right? Or in that direction. You can get a bit of a differentiation vector for every single cell. And here it's shown nicely on an example of neurogenesis in the mouse hippocampus, where you see these neuroblasts are kind of developing into these granule cells or CA1 or CA3 neurons. And you see the arrows follow very, very nicely this expected pattern. So we thought, you know, this is super cool, but at the moment, you cannot get this for spatial data, right? The reason is that most of the sequencing-based methods from which you can get these spliced and unspliced reads are not single cell resolution. And the imaging-based methods that are single cell resolution, they actually cannot detect these pre-mRNAs. Yeah? So we thought, you know, with SpaGE, we had the spatial transcriptomic data, we aligned it with the single cell data, we used precise, and then the data is aligned. So what we did is that we took from the single cell data also the spliced and unspliced counts, and we transformed them using the same alignment that we learned over here. And then we basically predicted these values for every spatial cell that we have from the spatial data. Yeah. So to test this, we looked at data that comes from the developing mouse brain. So this is a beautiful atlas, again, that comes from the Linnarsson lab. So this is all 10x single cell data. And for a specific time point, E10.5, they performed spatial transcriptomics using a protocol called hybrid in situ sequencing. So in this case, they measured the spatial expression of 119 genes. So we thought this is really nice. So we can basically integrate this with the single cell data from the same time point, and then transfer the spliced and unspliced reads from the single cell data to the spatial data, so we can get RNA velocities in the tissue itself. This is a UMAP of the spatial data. So every dot here should be basically a cell measured in the spatial transcriptomic data, and these are the velocities that we can infer. And of course, this is spatial data, so we can put every cell back to the XY location where it came from, right? And together with the arrow that we have learned. So this looks a bit messy, but so one example here is that this cluster 0, for instance, has no arrows at all going through it, which is not surprising. Cluster 0 is the mesenchyme layer that goes around the brain over here. So there is no interesting development of neuronal cells going in there. This is done shown at the cluster level, but we can zoom in at the single cell level. You get this picture that looks quite messy, but it's not very messy actually. So I highlight two examples here. So the first one is that if we look over here, these greenish cells, they are actually from this midbrain hindbrain boundary. So this is the hindbrain, this is the midbrain, and this is the boundary in between. And what you see here is that these cells are actually developing in 3 different directions. So those are becoming dorsal hindbrain, those are becoming dorsal midbrain, and those are becoming midbrain cells. So you see a bit this differentiation, but also in space. Yeah? There's another example here of these 4 brain cells that are developing into 3 different directions. So yeah, we're a bit excited about this. I think it's pretty cool to combine these differentiation trajectories with the spatial data. And here we show an example on brain data, but yeah, we're very curious to also apply this in other domains as well. So just to summarize the second part, yeah, using SpaGE, we can predict the expression of unmeasured genes at the single cell level, and we show that it slightly outperforms the other method, but it's much more scalable and flexible. So we can change the alignment method that we rely on quite easily, or the KNN imputation. The components of the method are quite easily adjustable. And we have now extended this to SIRV. So instead of only predicting gene expression levels, we can also go to other features, for example, RNA velocity. So going back to this full picture, I showed you that scHPL can address this part of this problem on integrating data that comes from different data sets. On integrating multiple modalities, we focused a bit on integrating spatial and single cell data. So I showed SpaGE and SIRV. I did not show anything over here, but this is a topic that we're really interested in. So for instance, how can you match genes across these different species? How does this affect your cell type matching after all? So if you call 2 cell types really homologous between species, and you then try to identify species-specific gene markers, is this really a marker, or did you actually match the wrong genes at the first level based on sequence similarity and not function? So yeah, this is something that I'm really interested in. So if you're interested in this, also let me know. And that's it. I do this work with a nice group of people. All the work I showed today, the scHPL was developed by Lika Michielsen, and SpaGE and SIRV were developed by Tamim Abdelal. And yeah, thank you for your attention.
Thanks a lot, Ahmed. The virtual applause is coming to you right now. Excellent, beautiful work. So questions,